So in mitosis, we start out with 23 pairs of chromosomes. For each chromosome, for each pair of chromosomes, one came from the mother of this male, okay, we're talking about the mitosis it's undergoing for typical body cells, and one came from the father. So whether we're looking at a epithelial cell, a muscle cell, or the spermatogonium, all of these cells are going to have 23 pairs. And I'm going to choose one of those pairs. So there's chromosome set 1, 2, 3, 4, 5, 6, 7, 8, all the way through 23. So total number of chromosomes equals 46. This is kind of like, do we start with the chicken or the egg kind of thing. So when the sperm fertilizes the egg, what we'll talk about in fertilization near the end of this unit, the sperm is going to be carrying 23 chromosomes, and the egg is going to have 23 chromosomes. So when the new cell is formed, we're back to 46. All right. This is a little easier to do with the sperm than it is with the egg, because we can look at the sex chromosomes, and within the sex chromosomes we have the X chromosome and we have the Y chromosome. So in mitosis, if we're making multiple spermatogonia, the chromosomes replicate, right? So we end up, as we move into prophase, that we have that's our X chromosome, our Y chromosome, and our X chromosome. Our new cell is going to have 2 X's and 2 Y's. Everybody with me so far? Okay, the nuclear membrane has broken down, and they line up along the equatorial plane of the cell, and we call that metaphase. And then microtubules our centrioles attach and shorten and pull these chromosomes apart. And so we end up in this cell with the X chromosome, in this cell with an X chromosome, in this cell with the Y, in this cell with the Y. And that would be the same for all the other 22 pairs of chromosomes. So we would have, obviously, the X is coming from the mother. But for chromosome 10, we would have a chromosome 10 from the mother and a chromosome 10 from the father. Right? Each of those chromosome 10s from the mother and the father would duplicate as sister chromatids. So the term sister chromatid is used here. And then those separate, and we have 2 new cells that look identical to the parent cell. So whether it's an epithelial cell undergoing mitosis, or whether it's a spermatogonium undergoing mitosis, each of these replicates and has identical daughter cells as far as the chromosomal material is concerned. This relationship, this pattern of chromosomal component, is known as a diploid cell. And that is classified as 2N, where in the human, N equals 23. Okay? So the spermatogonia cells undergo mitosis in the male over and over and over, making new spermatogonia. As we'll see next week in the female, that occurs as well with the oogonia, but that process stops before the girl is born. Okay, so there's no more mitosis occurring in the female in the ovaries after birth. Is everybody with me on this page? All right, now let's see what happens to the spermatogonia as we move into meiosis. The whole purpose is there's two things that become accomplished in meiosis. One is chromosomal number reduction. We go from a diploid cell to a haploid cell. That's the most important event that occurs. We don't want to fertilize, it would be non-viable if we had a new cell form that had 96 chromosomes. Or 90, I guess it would be 92 chromosomes. Right? The other thing that occurs, in addition to chromosome reduction, is exchange of chromatic material, what we call crossing over. Crossing over allows, let's say that in my egg, now I have two X's. One X came from the egg of my mother. The other X came from the sperm of my father. Okay. Now, in chromosomal reduction, those are going to be separated, and an egg that is fertilized would have only one X, either the one from my mother or the one from my father. For my children to get characteristics carried by the X chromosome, or if we're talking about chromosome 10, from both of my parents, that X needs to contain information from both my mother's X and my father's X chromosome. And that requires crossing over, exchanging of chromosomes between the two chromosomal types, between the two X's or between the two chromosomal 10s. So crossing over and reduction of chromosomal number is what is important in meiotic division. May I erase this board? Okay. Now we're looking at meiosis. Chromosome reduction. And crossing over. When I draw a cell, I'm really just drawing the nucleus. I'm not going to go through the nuclear membrane and all of that component. Okay? So we have a spermatogonia that's undergone mitosis. Its daughter cell over here is going to remain behind, capable of undergoing mitosis again and continuing to the population of new spermatogonia. Both of these cells are diploid, right? Both of these cells are 2N. Again, just like your epithelial cells, your nerve cells, muscle cells, and so on. See how many different colors I have. So let's follow two pairs of chromosomes. So this is a chromosome from the father. This is its paired chromosome from this guy's mother. Okay. So we'll say that's chromosome 2. Chromosome 2 from the father, chromosome 2 from the mother of this male. Then we'll have chromosome 22. So N is going to equal, because this was the only chromosome this cell had, N would equal what? looking at is this cell, not a human, okay? N would equal, how many different types of chromosomes do we have? 2. All right, we have the male and the 2 components from the parents of this cell, but N would equal 2. 2 times N is 4, okay? Now, the spermatogonium formed by this mitotic division looks just like the parent one. And now we're going to start meiosis. Meiosis is another cell division, and there are 2 stages of cell division in meiosis, not just one. Before we enter that first stage, we replicate the chromosomes again. So that's a set of sister chromatids of that chromosome. And that chromosome. And that chromosome. Okay. Notice cell division has not yet occurred. All we did was DNA replication, where we copied each chromosome, forming sister chromatids.
Excuse me.
As these are forming, there's a unique difference that occurs in meiosis as compared to mitosis. And that is these sets of chromosomes are attached to each other. So this set right here is going to be attached to its other pair. Remember, this came from the mother, this came from the father for chromosome 2. When they replicate, unlike in mitosis, they remain attached to each other. That's critical in meiosis. And this set is attached. These are known as tetrads. This close association in the tetrad formation is what allows crossing over to occur. So now, we don't have crossing over between these 2 sets, but between these 2. This is random, completely random. Kind of lie over each other, and sequences of nucleotides are cut, and the pieces of the chromosome are exchanged.
Bless you.
This cell is the primary spermatocyte. So when these changes, the spermatogonium starts to undergo these changes, it is now called a primary spermatocyte. So the first DNA replication, tetrad formation, and crossing over is what we identify as a primary spermatocyte. Notice that there is no cell division. A spermatogonium becomes a primary spermatocyte. It doesn't divide and form two primary spermatocytes. So it's the internal nuclear changes that differentiate a spermatogonium from a primary spermatocyte. Okay? Everybody with me on that? Now we're ready for our first meiotic division. By the way, when we go through this with the female oocyte, when a girl is born, all of her oocytes are as primary oocytes. They become arrested as a primary oocyte. And all through childhood, puberty, and into female reproductive active life, they stay as primary oocytes until they enter into the menstrual cycle. So you girls have women have a whole bunch of primary oocytes in your ovaries. And it doesn't actually complete cell division until just prior to ovulation. So each month, one or two maybe, oocytes will reach a secondary oocyte stage. Right? Otherwise, it might just shrivel up and die as a primary oocyte. And all the ones that are waiting for their turn to go through the cycle are still primary oocytes. Whereas when a boy is born, his cells are spermatogonia. And he continues to have spermatogonia as long as his testes are functional. Okay? Now, let's see what happens to the primary spermatocyte.